Hi students, welcome to SPSVERSITY. Today we are going to discuss about strategies for enhancement in food production. So why there should be an enhancement in food production? So what is the need for enhancement of food production? So here every year the increase in the population is increasing. So with the population increasing, the demand for food also increases. That is, we should increase the production of food also simultaneously. So where there is a necessity to increase the food production. To increase the food production, we apply various biological principles in animal husbandry as well as plant breeding to increase our food production. So nowadays, plant breeding, this animal breeding, this many techniques were developed like embryo transfer, tissue culture which is applied in plants, embryo transfer technology which is applied in animals, which play a pivotal role in enhancing food production. So, as the population is increasing, we need to go for the increase in food production. So, there are various sectors where we should increase the food production. So the first one is animal husbandry. So what is meant by animal husbandry? It is rearing of the animals like cows, buffaloes, these all will come. Not only rearing, breeding of animals, raising of livestock, taking care of livestock, not only cows and buffaloes, it includes poultry farming, that is hence fisheries, fish farming also. So in animal husbandry, along with breeding, we take care of livestock, the raising of livestock. It also includes, along with it includes poultry farming and even fisheries also. So first we'll see about dairy farming, dairying. So in this dairy farming, the management of animals, particularly for what purpose? Milk. So milk is an important, the nutrient one. So for which we will get many nutrients. So which is an important nutrient for human consumption. So dairy farming is mainly meant for increasing the milk production, that is for milk production. So in dairy farming, not only getting the milk, so the process as well as the systems that increase the yield and improve quality of the milk. So dairy farming means not only the obtaining of milk, to get quality milk as well as high yield of milk. Okay. So here when we come to dairy farming yield, okay. So here it is either cow or buffalo is raised, that is domesticated for getting clean milk. Okay. So here in this, to get a clean milk, there are various methods here. Like we should provide various facilities, the basic requirements, the food, water, the shelter, selection of the right breed of the buffalo, the transport of animals, daily care, feeding of the livestock, taking care of health, treating common diseases, the young one, the giving the young one, rearing the young one, management of waste, that is dung and urine, as well as reproductive management. So these all aspects are involved in the dairy farm management. So ultimately the product which we get is milk. Okay. So here when you come to the management of dairy farm, the first very important criteria is, so we should select good breeds, taking care of cattle, the buffalo and cow, and they should be given a proper shelter. So here they should be given a proper shelter, and they should be maintained disease free, and the quantity and quality of the fodder should be maintained. Clean lines and hygiene, that is the removal of dung, the urine from the poultry, from the dairy farm, it should be done. Regular inspection by veterinary doctor, that is keeping proper record keeping. Okay. So with the guidance of a veterinary doctor, they should be properly vaccinated. Treatment of diseases, vaccination, these all should be done to have a proper dairy farm management. Okay. Even when you come to poultry farm management also, that is the selection of breeds which are disease free, proper and safe farm condition, that is proper shelter, it should be provided, along with the food needs, that is proper feed and water, taking care of hygiene, health care. It also includes in poultry farm management. Okay. So here you can see the picture of a poultry farm where they are provided with feed, water, proper shelter, all the facilities. Okay. So here we are, when you come to dairy farm management and poultry farm management, we have the first very important criteria is we have discussed selection of proper breed. So what is meant by breed? So what is meant by breed? So a breed is a group of animals. By descent, they look similar in most of the characters, like their appearance, their features, size, configuration, etc. So a group of animals which has descended from, that is related by their common descent and similar in most of the characters. So that is called breed. Inbreeding, there are inbreeding, that is, as well as there is outbreeding. So what is meant by inbreeding? So breeding between animals of same breed for 4 to 6 generations. That means crossing the animals of the same breed for 4 to 6 generations. See here, if you take the example of just take the example of a dog, where these two were mated, that is sire and dam. Then here you can see it has given rise to the offsprings. In these offsprings, the offsprings which has come, that is they are mated among these offsprings, that is the male and female, they are mated. So this is inbreeding. Here also you can see the male and female of the offspring of the same breed which has come by crossing sire and dam, it is called inbreeding. So again it has given rise to the young ones. So this is called inbreeding. So what are the advantages of inbreeding? Inbreeding increases homozygosity. Homozygosity means both alleles are same. So when the both alleles are of same, it is called homozygous. So due to inbreeding, the homozygosity will increase. It leads to pure lines, helps in accumulation of superior genes, that is, which are of more productive. Elimination of less desirable genes, which are of not useful or less productive. So these are advantages of inbreeding. And there are disadvantages of inbreeding also. Like one major disadvantage is inbreeding depression. So what happens in inbreeding depression? When inbreeding is continued for more generations, that is continued close inbreeding for more generations. So what will happen when it is inbreeding is carried out for more generations? What will happen? It will reduce the fertility. Obviously, even it also reduces productivity also. So what is the solution for this? So the solution for this is animals can be mated with unrelated superior animals of same breed. That is going for outbreeding. Due to continuous inbreeding, it leads to inbreeding depression, which leads to reduction of fertility and the productivity. The next type of breeding is outbreeding. So outbreeding means breeding of unrelated animals. So here within the same breed but having no common ancestor. But in breeding there is a common ancestor for 4 to 6 generations. And it may be between two different breeds, or it may be between two different species of the same genus. So that means when you come to outbreeding, in outbreeding it may be of, outbreeding is of again, it may be of outcrossing, crossbreeding, outcrossing, crossbreeding, interspecific hybridization. So outcrossing means crossing of the individuals which has no common ancestor for 4 to 6 generations. Crossbreeding means crossing between two different breeds. Interspecific hybridization means crossing between two different species of the same genus. So first we'll discuss about outcrossing. Okay. So mating of animals within the same breed, but having no common ancestor. Okay, up to 4 to 6 generations. Having of no common ancestor up to 4 to 6 generations. Okay. So what is crossbreeding? Crossbreeding means it is crossing between two different breeds. Like, for example, if you take a sheep breed Husbandry, which is developed in Punjab by crossing between Bicanary Eves and Merino rams. So a cross is carried out between Bicanary Eves and Merino rams, which is combined, comes by, that is Husbandry. The new breed which has formed by crossbreeding, that is the Husbandry by crossing Bicanary Eves of sheep and Merino rams of sheep. What is meant by interspecific hybridization? It is crossing the male and female of two different species. Okay. Here if you take the female horse and male donkey. So the horse has 64 chromosomes and donkey has 62 chromosomes. That is, when a female horse and the male donkey is crossed, you will get an hybrid, that is the mule, which is sterile. So which is sterile. Okay. So it is called interspecific hybridization. Okay. So till now what we discussed is these all are conventional breeding methods where we cross between two breeds or two within the same breed without no common ancestor up to 4 to 6 generations and between two species of the same genus. As there is a development in the science and technology, a lot of new technology has come into breeding. Like one is artificial insemination. Okay. It is a controlled breeding experiment. To carry out controlled breeding experiment, we should carry out artificial insemination. So what is done in artificial insemination? The semen is collected from males and it is injected into the reproductive tract of selected female by the breeder. So that means here the semen which is collected by the male, it is injected into reproductive tract of female. And the semen which is collected, it can be stored or it may be used immediately and it can be stored at frozen state. Okay. The semen can be transported to long distances. There is no need to get the female. That is, the semen can be transported in frozen form to long distances where the female is present. So here, just carrying of semen is very easy than carrying of the female to the particular location. So in this, the semen is taken from the male and that is transported to the female where it is present. So it is a easy method where we can collect the semen and it can be injected into the female reproductive tract easily. So it is called artificial insemination, where the semen is injected into reproductive tract of the female. And next technology is MOET. What is MOET? MOET means multiple ovulation embryo technology. Multiple ovulation embryo technology. Okay. So this multiple ovulation embryo technology, what is the purpose of this? That is to improve successful production of the hybrids. So what is the process here done in multiple ovulation technology? So here the cow is, the female one is given it FSH. FSH means follicular stimulating hormone. So which will help in development of the follicles in female. So the follicular stimulating hormone is injected. Due to which what happens is the follicular maturation takes place and superovulation. Superovulation means more than one follicle matures at a time. Generally, one follicle will mature at a time during menstrual cycle. But here, due to FSH activity, follicular maturation more than one takes place. So more than one follicle means even the number of eggs also increase. That is 6 to 8 eggs will be produced instead of one egg per cycle. That means in menstrual cycle in 1 month only one egg is produced. But due to induction of this purposes 6 to 8 eggs, that means 6 to 8 follicles mature. Okay. And these eggs are collected. Okay. And they are, the female is either mated with the right bull either by naturally or artificial insemination. Okay. Once the male and female egg, or that is the male and female gamete, are fertilized, that is when the fertilization takes place. After that, we will get the fertilized eggs, which are of 8 to 13 cell stages. So these eggs are collected and they are transferred to surrogate mother. Surrogate mother means other than genetic mother. Okay. So the genetic mother is available for another round of superovulation. That means the genetic mother is used for superovulation. It is not used for pregnancy. The other are used for the pregnancy, that is, they are called surrogate mothers, where the pregnancy is continued. Whereas the genetic mother is available for next round of superovulation. So what is the purpose of this is it increases the head size. That means in one egg, when it is fertilized, naturally only one individual is formed. But here 6 to 8 individuals are formed. That means the head size will increase. Okay. So in this, another is the beekeeping. The beekeeping means the maintenance of honeybees. Okay. What purpose will maintain honeybees? That is production of honey. Okay. So honey is rich in high nutritive values and it is also good for medicine. Okay. Not only honey, we will also get bee wax, which is used in industries for preparation of cosmetics, polishes of various kinds. Okay. So beekeeping is practiced in where there are wild shrubs, fruit orchards and cultivated crops. So in India, the most common species is Apis indica, which is the bee, which is Indian bee, which is cultivated in India. That is Apis indica. Okay. To carry out successful beekeeping, that is to carry out successful beekeeping, we should have knowledge of nature and habit of bees. And we should select, that is, suitable location for beekeeping. That is fruit orchards, gardens. These are suitable places where lot of flowers are present, flowering plants are present, so that the beehives can collect nectar. So bees will collect the nectar. Okay. Catching and hiving of swarms, that is group of bees. So management during different seasons. Okay. Handling and collection of honey and as well as bee wax. Okay. Besides bees also help in plants, that is they are pollinators for many plants. Okay. Hence, beehives in crop fields, in flowering field increase pollination as well as they increase productivity, they improve honey yield. Okay. So beehives, that is bees, honey bees, not only help in giving the products, that is honey, as well as not only the bee wax, it also increases the pollination as well as the productivity of the plants also. Next one is fisheries. Okay. Okay. So when you come to fisheries is related to the catching, the processing and selling of the fish. Okay. So in this there is capture fishery as well as the fish farming. Capture fishery means where the catching of fishery, that is from the oceans, the rivers. Farming of fishes means in making in an artificial method. So here there are freshwater fishes like catla, rohu, common cod. Marine fishes, that is hilsa, sardines, mackerel and pomfrets. Okay. So here when you come to the culturing of these fishes, that is the pisciculture, which is called which comes under aquaculture. So the increase in the production of this fish production, it is called blue revolution. It is called blue revolution. And there is another revolution, that is green revolution. So what is meant by green revolution? So green revolution means it is increase in the plant production by applying plant breeding techniques. Okay. So here by green revolution we can increase the food production. Okay. As well as the more food production, even we can export it to other countries also. So here in green revolution, that is like the yield of many varieties, that is new varieties are developed in wheat, rice, maize, etc. by using this plant breeding techniques. So what is meant by plant breeding? So plant breeding is a purposeful manipulation of plant species. So manipulation means the changes. Okay. So to create the better suited for cultivation, as well as which gives high yield. So which gives high yield as well as disease resistant. So for what purpose will do plant breeding? That is for better cultivation, giving better yield, disease resistant. This all purpose we do plant breeding. Okay. So classical plant breeding just involves crossing and hybridization of pure lines, followed by artificial selection to produce plants. Okay. So when we come to the what traits generally we look into for plant breeding? To increase crop yield, improve quality, increase tolerance to environmental stress. Environmental stress means high salinity, high salt concentration, and to tolerate extreme temperature and drought. Drought means the arid conditions, and resistant to pathogens like diseases caused by viruses, fungi, bacteria, like many microorganisms. Increased tolerance to insects and pests. So these are the criteria which we take for breeding. Okay. The purpose of breeding, that is the traits which we take, is these all the traits which we take for breeding. So what are the different steps in plant breeding? The first step is collection of variability, evaluation and selection of parents, cross hybridization among the selected parents, selection and testing of superior recombinants, testing and release of commercialization of new cultivars. Cultivars means the seeds of new varieties. So welcome to SPSVERSITY. Follow us on the social media. So first we'll discuss about collection of variability. What is this collection of variability? So in any species there will be, that is, wild varieties. So in this collection of variability, the collection and preservation of different wild varieties, this collection is an important step. After collection, evaluation for their characteristics. Whatever the wild varieties we collected, they should show the desirable features. Desirable features means they should be resistant, disease resistant, environmental stress resistant, they should give high yield, insect and pest resistant. So whatever the wild varieties are collected, so in these wild varieties, all the alleles of all genes are collected. So this is called germplasm collection. So germplasm means the genes, that is alleles for all genes, it is called germplasm. So where it is collected is from wild varieties. Okay. After the collection of variability, the next step is evaluation and selection of parents. So whatever the germplasm is collected, it is evaluated for desirable combination of characters. So desirable combination of characters means the resistance, capability to give high yield. These all are desirable characters. So when they show desirable characters, then they select the plants, and these selected plants are multiplied. Multiplied means they will increase the number of selected plants. And later on, these after increasing, they're subjected to inbreeding. So inbreeding leads to evaluation of, that is, pure lines. Okay. So once the desirable plants are selected, then they are multiplied and subjected to inbreeding. So inbreeding leads to formation of pure lines. So these pure lines are created before going to the artificial cross hybridization. These pure lines which are formed by inbreeding, they are subjected to cross hybridization. So the two selected parents are subjected to cross hybridization. So in this cross hybridization, whatever the plant we take, the female, it is subjected to emasculation. Emasculation means removal of stamens from the female flower to prevent self-pollination. Next, bagging, that is the covering of female flower with a bag, that is called the female, that is bagging, to prevent undesirable pollination. Okay. So here, for the female plant, these processes are carried out: emasculation, bagging. Okay. So for male plant, that is whatever the male plant we take, we don't do any process. There is no need of process. Just collection of the pollen from it and dusting on stigma of the female. Okay. So here, these which we get, that is the plants which we take. For example, if we take one parent is of high protein quality, it may not be having the disease resistance. That means other parent should be disease resistant. So when you hybridize these two, we'll get the both characters, that is both high protein quality and disease resistance. Okay. So in hundreds of the offsprings, only one or two will show the desirable combinations. So for female flower, the emasculation and bagging is done. So once it is done, that is after the pollen grain are collected and dusted on the female stigma. Okay. So once the pollination is done artificially, the fertilization takes place. After pollination, the next process is the fertilization. In the process of sexual reproduction, fertilization, which leads to formation of seeds. Okay. Once the young one, that is the progeny, are formed, okay, from this progeny, again this selection is done in progeny. Okay. So for this selection, it requires careful observation and evaluation of progeny for desirable characters. Okay. In this, the superior plants are selected. Okay. And these hybrids which are selected, they are self-pollinated, which leads to the pure lines. So again, they are subjected to self-pollination. Again, they do for the cross hybridization. So this process is continued for 4 to 6 generations. The same process: evaluation of progeny, selection of the desirable progeny, After selection of desirable progeny, the next one is they are subjected to, that is inbreeding. Again they are done to cross hybridization. That is, this is continued for 4 to 6 generations. Okay. So after 4 to 6 generations, after getting the one which give high yield, the agronomic traits, that is the quality yield, disease resistance. So once the evaluation is done, so evaluation means like recording their performance under ideal fertilizer, irrigations, under ideal conditions. Then it is sent for testing in the former fields, at least for 3 generations. Okay. Then it is evaluated, the data is evaluated with the best available one. So the best available one is multiplied and released into the market. That is, it is such a long process where a lot of process is there, where the cross hybridization, the selection, evaluation, these all are done for many generations. By then we'll get a superior variety which is suitable to the cultivation in the local crops. Okay. So this is what in plant hybridization is done. Follow SPSVERSITY on social media. So here when you come to wheat, in green revolution there is a lot of varieties has come in wheat, rice, maize, etc. So here for green revolution, Nobel laureate, that is Norman E. Borlaug at International Center for Wheat and Maize Improvement in Mexico, he has developed semi-dwarf varieties of wheat. Whereas when you come to India, in India a lot of high-yielding varieties of wheat are produced, that is Sonalika and Kalyan Sona, which are wheat varieties which are developed in India. So which are wheat varieties which are developed in India. Come to rice. So semi-dwarf varieties were developed, that is IR8 is developed in International Rice Research Institute, which is located in Philippines. And for in Taiwan, there is a Taichung native variety, that is TN1, which is developed in Taiwan. Okay. In India, Jaya and Ratna are semi-dwarf varieties which are developed in India. Okay. So these are Jaya and Ratna. Whereas wheat, Sonalika, Kalyan Sona are wheat varieties which are developed. Along with rice and wheat, even other are also developed, like sugar. Sugar cane, that is in North India, that is the Saccharum barberry of North India, which has poor sugar content. It is crossed with Saccharum officinarum, which has thick stems and high sugar content. So by the end we get, that is with high sugar cane content, thick stems. That is high sugar and thick stems. So now when there is a cross done between Saccharum barberry and Saccharum officinarum, we got a variety of sugar cane which is high in sugar with thick stems. Okay. So till now what we discussed is, that is the increase in yield. Now that is development of disease resistant is also an essential aspect. Why? Because lot of fungicides, insecticides, lot of things are sprayed. That is, these fungicides and these are very expensive one. The cost of agriculture increases. At the same time, there is also loss in agriculture due to these diseases. To prevent this loss and the expenses, to cut off the expenses, we should develop varieties which are resistant to diseases. Like for example, if we take generally in plants, fungal diseases are brown rust of wheat, red rot of sugar cane, late blight of potato. Bacteria, black rot of crucifer. Viral diseases, tobacco mosaic, turnip mosaic, these are all bacterial diseases. viral diseases. Okay. So how to develop disease-resistant varieties? So what is the method for this? That is, the method is the screening of germplasm for resistant resources. Okay. So hybridization of selected plants. First the screening process, next hybridization. Selection and evaluation of hybrids. Testing and release of new varieties. Okay. So in this, to develop the new varieties, that is disease-resistant varieties. So we use a type of breeding, that is a developed method of breeding, that is mutation breeding. So here, mutations are sudden changes which are seen in the living organisms, which may be beneficial as well as harmful. That is, changes in the genetic material, genetic variability by mutations. How can they be induced? By applying mutagens. Mutagens means the substances which cause mutation. It may be chemical or physical. So in this also, the first screening of the parents, the selection of parents with desirable characters, okay, which are developed by mutation. Okay. So here, in this, lot of varieties are developed, like mung bean, which is resistant to yellow mosaic virus, and as well as powdery mildews, which is resistant to powdery mildews, which are developed by mutation breeding. So one of the varieties, that is Parbani Kranti. The variety name is Parbani Kranti, which is a variety of bindi, lady's finger, abelmascus esculent, which is resistant to yellow mosaic virus. Okay, so it is actually, that is, it is a variety of wild species, a new variety that is developed, that is Parbani Kranti, which is resistant to yellow mosaic virus. Okay. Naturally, in plants, there are characters which will resist the entry of the insect and pest. So in some plants, we can see hairy leaves which will prevent the entry of pathogens. Solid stem in wheat leads to prevention of stem sore fly. Smooth leaves and nectarless cotton variety do not attract bollworms. High aspartic acid and low nitrogen sugar content, which in maize, it will be resistant to stem borers. So that is, in plants, naturally there are some adaptations to prevent the entry of pathogens, that is insects and pests. Okay. So to protect this insect and pest, that is, even there is a development in plant breeding to have insect and pest resistant varieties. So lot of insect and pest resistant varieties are developed in plants. Okay. Not only disease resistant, as well as the insect and pest resistance are also developed. Okay. Now we will discuss about hidden hunger. Follow SPSVERSITY on social media. So what is meant by hidden hunger? Hidden hunger means we'll have the proper quantity of the diet, but we don't get the quality. So that means suffering from micronutrient, protein and vitamin deficiency. Even though we get the quantity of the food, when you take in the terms of quantity, it is very good. But when it comes to terms of quality, it is of low quality. So when they get low quality of the food, that is, which is deficient in vitamins, proteins, nutrients, they will suffer with many disorders. So it is called hidden hunger. Okay, hunger means lack of proper food, lack of food. But here, hidden hunger, it is a quantity wise it is good, but quality wise it is less. So when there is no proper nutrients, what will happen? Like there are many nutrients which are required, like iron, zinc, vitamin A, iodine, zinc. These all increase when lacked in the diet. They will increase the risk of disease. They will reduce the lifespan, which leads to Unstability is psychologically, that is reduce mental ability. Okay. So what to do to increase the quality of the food that is rich in the nutrients? So that is called BioFortification. So what is done in BioFortification? So in BioFortification, crops are breeded for higher level of vitamins, minerals and higher proteins and okay. So that the public health will improve. So when the public health improves, that is, the occurrence of diseases will decrease. So lot of diseases nowadays, the public are suffering with a lot of deficiency diseases, vitamin deficiency diseases, mineral deficiency diseases. These all can be reduced by BioFortification. So they all can be reduced by BioFortification. So it is a breeding of crops with a higher level of vitamins and minerals. Okay. So the objectives of BioFortification are to improve varieties with high protein content and quality, oil content and quality, vitamin content, micronutrient and mineral content, so that all the people will get all the nutrients, the minerals, the vitamins, so they don't suffer from the deficiency disorders. Like hybrid maize was developed with twice the amount of amino acids, lysine and tryptophan when compared with maize, which are very important required for human development. Variety, that is wheat variety at class 66, which has high protein content. Iron fortified rice, that is, which has more iron than this. And there is a golden rice, which is rich in vitamin E. Nowadays we are getting golden rice, which is rich in vitamin E. So like this, there are a lot of varieties are developed where they are rich in protein content, the vitamins, the minerals which are required for the humans. So when we come to Indian Agricultural Research Institute in New Delhi, which has developed vitamin A enriched carrots, vitamin C enriched bitter gourd, mustards, tomato, iron and calcium enriched spinach and butterworm, protein enriched broad, that is lab love, French beans and cotton peas. So like that, there are a lot of varieties which are developed which are rich in the nutrients, the protein, the vitamins, the minerals. Okay. So besides all these, the plant breeding, the disease resistant varieties, the insect and pest resistance, all these, there is an alternative for increasing food production. That is single cell protein other than plants and animals. Okay. It is an alternate source for proteins. Okay. So in this single cell protein, what actually they do is they cultivate microorganisms at low cost. Okay. So it is an alternative source for protein. For example, if you take a green algae spirulina, which can be grown on wastewater coming from potato processing plants, straw, molasses coming from sugarcane industries, animal manure, even sewage. Okay. So not only spirulina, there are other species also like chlorella, which is rich in protein, and algae, which is rich in protein. So like this, there are many algae which are rich in proteins, which are used as a source of proteins. So why we are using microorganisms source of protein? What is the reason behind this? That is, for example, if you take a 250 kg cow, per day it gives 200 grams of protein per day. Okay. See the ratio, 250 kg cow gives 200 grams of protein. But at the same time, if you take 250 grams of a microorganism, that is a bacterium Methylophilus methylotrophus, which gives 25 tons of protein. See the ratio, 250 kg cow, 200 grams of protein. 250 grams of microorganism, 25 tons of protein. Not only even mushrooms, is also a fungi, which is a microorganism which will produce, that is this one. So single cell protein, that is generally we think that single cell microorganisms are used. Not only single cell, it is a misnomer. That is not only single cell organism or microorganisms, even multicellular also are used. Okay. The next alternative that is to increase the plant production is tissue culture. So what is tissue culture? It is done artificially to increase the number of plants in a short span of time. So in tissue culture, there are main aspects like totipotency, explants, micropropagation, somaclones. We'll see here. So when you take a plant cell, each plant cell is capable of forming the entire plant. That is called totipotency. So every plant cell has the capacity that is to develop into entire plant. That is called totipotency. So here you can see a small tissue of a carrot, that is a small tissue of carrot is taken for tissue culture. So this small tissue, that is from where it is taken, that is, it is called explant. So explant may be a root, stem or a fruit or whatever you call. So this a tissue which is kept in nutrient medium. So this is a flask that is which is kept in a nutrient medium. So when it is kept in nutrient medium, after 2 to 3 weeks, it develops into an callus. So from callus, that is, the new plantlets are formed. So here you can see the plants are potted. Later on, they are shifted to the soil, natural soil. So here in this, from one explant, if you observe very carefully, we are getting a lot of plants. Okay, generally from one seed we will get only one plant. But here, from a small tissue, we are getting a lot of plants. That is, within a short span of time, within a short space, we can increase the plant production, which is called the micropropagation. So which is called the micropropagation. Okay. And here, due to this, within a short span of time, a lot of plants can be produced. And even we can see here variations, they are called somaclonal variations. Okay. In this tissue culture, there are different types of tissue culture like embryo culture, okay, ovule culture, ovary culture, okay, meristem culture is there, which is meant for virus-free plants. To get virus-free plants, we use meristem culture. Okay. So here there are a lot of applications of tissue culture. That is, large number of plants can be produced from small tissue or from a single cell. Genetically identical plants are produced, which are called somaclones. And even healthy plants can be produced, that is, virus-free plants can be produced by culturing meristems. By culturing meristems means the meristems, then tissues are kept in tissue culture. Okay. Why? Because the meristems are free of virus, they will multiply rapidly than the virus. Okay. These are applications of tissue culture. So tissue culture is another method to increase the number of plants within a long short span of time. Okay. And there is somatic hybridization. What is meant by somatic? Somatic means crossing two normal vegetative cells. Okay. So in this somatic, when you come to the process, the first cells are isolated from the plants, from the desirable plants. The cell wall of the cells is digested by using enzymes, cellulase and pectinases. When cell wall is digested, whatever it is left out is called protoplast. The two protoplasts of different plants, that is desirable plants, are crossed, that is, they are fused to form an hybrid protoplast. To fuse with protoplast, they use various methods like using electric field or by adding polyethylene glycol. And the hybrids which are obtained by this, they are called somatic hybrids, and this process is called somatic hybridization. So here when there is a fusion of protoplast of potato and tomato, it has lead to, that is, pomato, a variety called pomato. That is, so this is pomato. From the ground in the inside the soil we can get the potatoes. Okay. From the top, that is above the soil, we can get tomatoes, which is called variety, but it is not successfully, economically it is not successful, but it is a good initiative for somatic hybridization. So nowadays, lot of varieties are developed like this by using somatic hybridization. So single cell protein is meant for alternative protein source. Tissue culture is meant for increasing the number of plants. Somatic hybridization, it is meant for crossing between two different species of plants, like tomato, which can be crossed with potato, which will get tomato, which they will use both potato and tomato. So like this, these are different methods to increase the food production, like the hybridization in animals, that is the breeding in animals, plant hybridization, mutation breeding, breeding for disease resistant, breeding for insect and pest resistant. Next, that is the BioFortification, the hidden hunger, the single cell protein, and tissue culture. So these are the aspects by which we can increase the food production. So thank you all. We'll meet in the next session. Welcome. Follow SPSVERSITY on social media.